The overall goal of this strand-specific primer extension qPCR assay is to quantitatively assess the repair of adducts cross-linked to plasmid DNA following transfection into mammalian cells.
This method can help answer key questions in the DNA repair field, such as what pathways are involved in the repair of DNA-protein crosslinks. This technique has the potential to provide a better understanding of the DNA damage response, because it permits one to selectively study the repair of DNA-protein crosslinks in the absence of other types of DNA damage. Though this method can provide insight into the repair of DNA-protein crosslinks, it can also be applied to other types of DNA damage, such as abasic sites and other polymerase-blocking adducts. The main advantage of this technique is that it can detect repair of adduct-containing plasmids at time points as early as 2 hours.
We initially intended to use host cell reactivation to study repair. However, those assays don't directly measure repair and may overestimate repair efficiency, particularly if RNA polymerase can read through incomplete repair products. Visual demonstration of this method is critical as the purification and strand-specific primer extension steps are otherwise difficult to visualize.
Mix 20 microliters of a solution containing 80 picomoles of an 8-oxoguanine containing oligonucleotide with 5 microliters of 10x ligase buffer. And add 1 microliter of a solution containing 10 units of T4 polynucleotide kinase. Adjust the final volume to 50 microliters and incubate the tube in a water bath for 30 minutes at 37 degrees Celsius. Next, constitute the primer extension reaction on ice.